If you were casting the ultimate Hollywood blockbuster about the human body, um, for decades, the medical community basically treated cholesterol like it was Thanos.
Right, just the ultimate inevitable supervillain.
Exactly. The Thanos of the breakfast table, just ready to snap its fingers and wipe out your cardiovascular health with a single plate of bacon.
It is a great comparison because, honestly, the reality of human biology is a lot more nuanced than a comic book movie.
Yeah, what if cholesterol isn't Thanos at all? What if it's actually more like Alfred to our body's Batman?
I love that, the unsung hero.
Right. Quietly working in the shadows, patching up the damage, managing the biochemical bat cave, and literally keeping the hero alive. Welcome to the Deep Dive Lab.
It really is a perfect premise to start with, especially for a topic that affects absolutely everyone listening to this right now. We're dealing with a molecule that is fundamentally misunderstood by the general public,
And misrepresented, honestly, by parts of the medical establishment for a really long time.
without a doubt.
And that is the core mission of our deep dive today. We are going to completely demystify cholesterol. We want to transform it from this feared buzzkill medical word into what it actually is.
Which is a fascinating, essential, and highly dynamic building block of human life.
Spot on. And we have a fantastic stack of sources for today's deep dive to help us reframe this conversation. We're looking at historical texts, cardiovascular guidelines, and some truly groundbreaking research papers.
Yes, from journals like the Journal of Alzheimer's Disease Reports, the Journals of Gerontology, and the Journal of Lipid and Atherosclerosis.
Okay, let's unpack this. Where does the story of this misunderstood master builder actually begin?
Well, the initial discovery happened in a rather unglamorous place.
Right.
Back in 1769, a French researcher first isolated it from human gallstones.
Gallstones. Glamorous.
Very. And it basically sat around as a biochemical curiosity until 1815, when chemist Michel-Eugène Chevreul rediscovered it and named it cholesterine.
Cholesterine, which comes from Greek, right?
Right. From the Greek word chol for bile and stereos for solid.
Yeah.
Later, they added the old suffix because it's technically an alcohol.
Solid bile alcohol. I mean, I can see why the marketing department decided to just stick with cholesterol.
Yeah, much catchier.
But let's fast forward past the 18th century gallstones, because the modern scientific obsession with this molecule is what's really staggering.
Staggering is the right word. Scientists have been completely obsessed with this molecule for centuries.
Over 10 Nobel Prizes are directly or indirectly related to cholesterol and lipid research. You don't hand out 10 Nobel Prizes for something that just sits in your arteries causing trouble.
You certainly don't. The scientific community recognized early on that without cholesterol, human biology simply ceases to function.
Just stops.
Completely. To give you an idea of the scale, a typical 150-pound human male manufactures about 1 gram of cholesterol every single day.
Mostly in the liver and intestines, right?
Correct. And stores around 35 grams in the body at any given time. The body guards this supply incredibly closely.
Which brings us to one of those Nobel Prizes, right? The 1985 prize.
Yes, awarded to Brown and Goldstein. They won it for discovering how tightly the body controls cholesterol through something called the SREBP pathway.
SREBP. Let's break that down for everyone listening, because this is where the biological engineering gets really cool.
It really does. So SREBP stands for sterol regulatory element binding proteins. You can think of it as a microscopic cellular thermostat.
Okay, a thermostat.
Right. These proteins sit on the membrane of the endoplasmic reticulum inside your cells. When they sense that cellular cholesterol levels are dropping too low, the SREBP protein physically detaches.
It just breaks off.
It breaks off, travels into the cell's nucleus, and binds directly to the DNA. It essentially flips a genetic switch,
Telling the cell to do what?
telling it to manufacture more cholesterol receptors, to pull more in from the bloodstream, or to just synthesize more of it from scratch.
So it's an automatic, built-in feedback loop. The body is constantly monitoring the thermostat.
Constantly. And it has to, because cholesterol is the literal physical scaffolding of our cells.
This is where I like the bouncy castle analogy. Think of your cells like a microscopic bouncy castle.
That is a great visual.
You want the walls of that bouncy castle to be flexible enough that they don't shatter into a million pieces when someone jumps into them. But you also need them to be robust enough that they don't just melt into a puddle.
Exactly. Cholesterol is the structural framing of that bouncy castle. It is the Goldilocks molecule for your cell membranes.
The Goldilocks molecule, not too hard, not too soft.
Right. Because it has a rigid ring structure but a flexible tail, it wedges itself right between the phospholipids in your cell membranes. At lower temperatures, when the membrane wants to pack tightly together and freeze into a rigid ice block, Cholesterol pushes the molecules apart,
Keeping it fluid.
keeping it fluid.
Yeah.
But at higher temperatures, when the membrane wants to become too fluid and fall apart, cholesterol acts like a biochemical glue holding it together.
So it's keeping us from freezing or melting on a cellular level, but it's also a chemical precursor, which connects right to your everyday life.
Absolutely.
If you are listening to this right now, cholesterol is literally the molecule that got you out of bed today and is helping you handle the stress of your daily life.
It is the foundational raw material. Within your cells, cholesterol is converted into vitamin D when sunlight hits your skin.
Which is huge for immune health and mood.
Huge. And the liver uses it to synthesize bile acids, which you absolutely need to digest dietary fats.
Right.
And crucially, it is the parent molecule for all your essential steroid hormones. We were talking about cortisol, which manages your stress response, and aldosterone, which regulates your blood pressure.
And the sex hormones too. Testosterone, estrogen, progesterone, they all start as a cholesterol molecule.
They do.
Which really makes you think. When we historically declared this total blunt force war on cholesterol, we were inadvertently declaring war on our body's ability to handle stress, build strong bones, and regulate reproduction.
That is the real danger of oversimplifying complex biochemistry. We turned a vital building block into a villain.
Yeah.
And that danger becomes even more apparent when we look at how the brain handles cholesterol, because the brain's landscape is completely isolated from the rest of the body.
Oh, here's where it gets really interesting.
It does.
The brain only accounts for about 2% of your body weight, but it hoards roughly 20% of your body's total cholesterol.
It is incredibly greedy with it.
It uses it to form myelin sheaths, right? The protective insulation around nerve fibers that lets electrical signals fire rapidly.
Exactly. But the catch is, the brain doesn't just order a cholesterol delivery from the liver.
It can't.
It absolutely cannot.
Ah.
The blood-brain barrier is highly restrictive. Peripheral cholesterol from the blood simply isn't allowed to cross that barrier.
The brain is basically an ultra-exclusive Hollywood VIP club. The blood-brain barrier is the big, intimidating bouncer at the door.
I like that.
And all the peripheral cholesterol floating around in your blood, that's just paparazzi waiting outside. It's not on the guest list. It can't get in.
So how does the VIP club get its drinks?
Right. It turns out the brain has its own internal bartenders. There are these specialized cells inside the brain called astrocytes.
Astrocytes.
Yeah, and these astrocytes manufacture cholesterol internally, package it up in a specialized brain-only format, and slide it directly over to the neurons that need it.
What's fascinating here is the elegance of that isolated system.
Mm-hmm.
But it begs the question, what happens when that system is breached?
The paparazzi storm the club.
Exactly. And this brings us to a really compelling new hypothesis for Alzheimer's disease.
Right, from the Journal of Alzheimer's Disease Reports.
Yes, authored by Jonathan Darcy Rudge from the School of Biological Sciences at the University of Reading in the UK. He introduces a concept called the lipid invasion model.
The lipid invasion model. It sounds like a sci-fi thriller. Let's unpack the mechanics of this because it challenges a lot of what we thought we knew about dementia.
It really does. Traditionally, Alzheimer's research has focused heavily on the accumulation of amyloid beta plaques and tau tangles in the brain.
The plaques were always the bad guy.
Always. But Rudge's hypothesis looks upstream of those plaques. His model suggests that due to aging, physical trauma, or chronic vascular disease, the tight junctions of the blood-brain barrier begin to degrade.
The bouncer falls asleep.
The bouncer falls asleep. And when that happens, external lipids and peripheral cholesterol from the bloodstream invade the brain's VIP section.
So what does that actually do to the neurons? It's just too much cholesterol.
It creates total chaos. The astrocytes, your bartenders, are used to tightly regulating a pristine environment. When peripheral cholesterol floods in, it disrupts the brain's delicate internal lipid balance.
Wow.
And here's the twist. The current thought is that the production of amyloid beta plaques might actually be a misguided defense mechanism by the brain.
An emergency response.
Yes, an emergency response to this foreign lipid invasion. The plaques are the debris of a broken system, not necessarily the root cause.
That completely changes the narrative. We've spent decades trying to clear the amyloid plaques, but this model suggests the plaques are just the smoke. The fire is the breakdown of the blood-brain barrier and the invasion of outside cholesterol.
Precisely. And this isn't just a theoretical model in a vacuum. We are seeing real-world, long-term tracking data showing how cholesterol behavior over a person's lifespan predicts these neurological crashes.
This connects perfectly to the study by Adina Zeki Al-Hazuri and her team from Columbia University.
Yes, published in the Journals of Gerontology Series A.
Biological Sciences and Medical Sciences.
That's the one. And this study provides a crucial longitudinal piece of the puzzle. They didn't just look at a single blood test.
Right, they looked at 20-year lipid variability.
Throughout young adulthood and midlife. And then they correlated that variability with midlife cognitive function and structural brain integrity.
The findings there are a massive warning sign. For decades, you go to the doctor, they look at your chart and say, is your cholesterol high or is it low?
A simple snapshot.
Right. But Al-Hazuri's team found that the absolute number isn't the only thing that matters. It's the fluctuation.
The bouncing around.
Exactly. If your cholesterol levels are bouncing around from year to year over decades, high one year, low the next, then high again, that yo-yo effect is heavily associated with the decline in cognitive function and physical damage to the brain's white matter.
So what this means for you, the listener, is that consistency is key.
Why does variability cause so much damage, though, mechanistically?
The prevailing theory goes right back to the blood-brain barrier. It's about endothelial dysfunction.
Endothelial dysfunction. We hear that thrown around a lot in cardiology. What is physically happening to the blood vessel when lipids fluctuate like that?
Imagine taking a piece of fine sandpaper to the inside of a garden hose.
Ouch. Okay.
Consistent lipid levels, even if they are slightly elevated, allow the endothelium, the inner lining of your blood vessels, to adapt and maintain its structural integrity.
It gets used to the pressure.
Right. But wild fluctuations trigger an erratic inflammatory response. The endothelial cells become stressed, they lose their elasticity, and the tight junctions holding them together start to pull apart.
And because the blood-brain barrier is made of these exact endothelial cells,
When they pull apart, the barrier becomes permeable.
the garden hose leaks, the bouncer fails, and the brain gets flooded. The lipid invasion model.
Exactly. So yo-yo dieting, constantly starting and stopping lipid medications, or adopting extreme lifestyle fads that cause your cholesterol to swing wildly.
You might be actively wearing down your blood-brain barrier. Your brain craves consistency.
It does. It requires us to move away from that snapshot approach to medicine. A single fasting lipid panel tells us very little. We need to look at the 20-year trend line to truly gauge dementia risk.
Since we are talking about the long-term trend line and diets, let's talk about the diet drama.
Well, the diet drama.
Because the dietary guidelines for cholesterol have had more confusing reboots than the Spider-Man or X-Men movie franchises.
They really have.
In the 80s and 90s, an egg yolk was treated like a ticking time bomb. But then, the 2015 dietary guidelines actually dropped the strict 300 milligram per day limit on dietary cholesterol. If our blood vessels are so sensitive, why did the medical community backpedal on eggs?
They backpedaled because the science finally caught up to the biological feedback loop we discussed earlier, the SREBP pathway.
Right, the thermostat.
For a long time, the assumption was strictly linear. If you eat more cholesterol, you will have more cholesterol in your blood.
Makes logical sense on the surface.
It does, but human metabolism is far more elegant than a simple plumbing pipe. If you eat more cholesterol, like a plate of eggs, your liver usually just makes less to compensate.
But wait, if the liver naturally downregulates production when we eat dietary cholesterol, how did the entire medical community get it so wrong for 40 years? How did we end up choking down egg white omelets for decades?
They got it wrong because of the trap of observational epidemiology. Historically, researchers would look at a large population, ask them what they ate via food frequency questionnaires, and see who had heart attacks.
Where people notoriously lie about what they eat.
They do, and those studies are riddled with confounding variables. A person who ate a lot of eggs in the 1980s was likely eating them alongside bacon and sausage.
Cooking them in trans fats.
Exactly. And perhaps generally ignoring health advice, which might mean they smoked or didn't exercise.
So it's just noise. You can't isolate the egg from the bacon or the egg from the cigarette.
Precisely. Observational diet studies can only show correlation, never actual causality. So to figure out what actually causes atherosclerotic cardiovascular disease now, we needed a better tool.
Which brings us to the research by Dai Sitko from the Gyecheon University College of Medicine.
Yes, published in the Journal of Lipid and Atherosclerosis. Code details the impact of something called Mendelian randomization.
Mendelian randomization. It sounds incredibly dense, but the underlying concept is brilliant. Mendelian randomization is basically nature's own A/B testing.
It is a phenomenal genetic tool. Think about a traditional randomized clinical trial. Researchers take thousands of people and randomly assign half to a drug and half to a placebo.
Right.
That random assignment distributes all those confusing variables like who exercises and who eats bacon equally across both groups. But we cannot ethically randomize people to eat a high cholesterol diet for 30 years to see if they die.
Right. But Mendelian randomization says we don't have to do that because nature already randomized us at birth.
Exactly. When you are conceived, you randomly inherit genetic variants from your parents. Some of these tiny variants naturally dictate that your liver will produce slightly more LDL cholesterol over your lifetime.
The bad cholesterol.
Right. Other variants dictate you will produce slightly less.
So researchers can look at massive databases of hundreds of thousands of people, group them by these random genetic lottery tickets, and see who developed heart disease.
Yes, and because these genes are assigned at birth, they don't change based on your environment. Your genes don't care if you lie on a dietary questionnaire.
That is the magic of Mendelian randomization. It cuts right through decades of lifestyle noise.
It does. And using this method, researchers definitively proved actual causality between lipid fractions like LDL and heart disease. It is no longer just a debate or an association.
We know for a biological fact that cumulative, lifelong exposure to high levels of circulating LDL cholesterol directly causes the buildup of plaque in the arteries.
Yes. It's not necessarily the eggs. It's your genetic lottery dictating your biological transport system.
And these genetic insights aren't just for publishing papers. They lead to some incredible future tech. Once we identify the specific genes causing the logistical breakdown, we can build tools to fix them, like the PCSK9 gene mutation.
The PCSK9 discovery is a watershed moment in personalized medicine. Through genetic tracing in people with familial hypercholesterolemia, dangerously high cholesterol scientists identified this specific gene.
What does it do?
PCSK9 produces a protein that binds to the LDL receptors on your liver. Those are the very receptors that pull bad cholesterol out of your blood, and it marks them for destruction.
So if your genetic lottery ticket makes you produce too much PCSK9, you destroy your own liver receptors. Your blood cholesterol skyrockets, scraping up the endothelium.
Exactly. But because we figured out that exact mechanism, we now have incredible new therapies that can save lives.
Therapies that sound like they belong in a sci-fi movie.
We have PCSK9 inhibitors now, which are injectable antibodies that seek out and neutralize that specific protein. It preserves your LDL receptors and drastically lowers bad cholesterol.
And there's even CRISPR, right?
Yes. There are human trials underway using CRISPR gene editing technology to go into the liver cells and permanently turn off the PCSK9 gene.
They are literally editing the genetic code to permanently fix the transport system. We are moving from blunt force guidelines like don't eat eggs to highly personalized molecular precision medicine. It's astonishing.
It is a remarkable evolution.
So let's pull all of these threads together. What are our key takeaways from this stack of research today? First, cholesterol is a vital Nobel Prize-winning building block of life, not just a breakfast villain.
You couldn't survive without it.
Second, the variability of your cholesterol over time is a crucial, newly discovered warning sign for brain health and cognitive decline.
Protect your blood-brain barrier.
And third, genetic tools like Mendelian randomization are cutting through decades of diet confusion to pave the way for highly personalized, sci-fi-level medical treatments.
If we connect this to the bigger picture, the ultimate takeaway here is recognizing the biological paradox of cholesterol.
The paradox.
Yes. It is the molecular spark plug for our hormones and the architectural foundation of our brain function. It only becomes our greatest adversary when our body's logistics and delivery systems break down.
Due to genetics or chronic stress.
Right. The molecule itself isn't evil. It is the environment it finds itself in that dictates health or disease.
Thank you so much for joining the Deep Dive Lab today. If you loved uncovering the truth about the body's ultimate unsung hero, please hit that subscribe button on the channel so you never miss a discovery.
And before you go, consider this. We now know that the brain manufactures its own highly exclusive, isolated supply of cholesterol via those astrocytes.
Right, the brain's VIP bartenders.
So here is the thought to chew on.
Since the lipid invasion model points to a barrier breakdown, could the ultimate cure for neurodegenerative diseases lie not in clearing out plaques from the blood, but in figuring out how to convince the brain's VIP bartenders to change their recipe? Keep diving deep, and we will see you next time.